Thank you so much for inviting me, Sabith. I really appreciate it. I think it's very important to have collaborations and meet some new people that I probably otherwise would have not met. One of the things that I want to mention is the this preliminary work. I'm presenting two projects here, but the big project that I'm presenting is comes from NASA, and then how that has created another project later on. So all of that comes out of the necessity that NASA has for humans landing on the moon and going into deeper space. And also in extreme environments and how we're going to deal with the highest risks that there are. My background is I am a physician, and so the most important thing for me is crew safety, but that doesn't mean that is the only goal, right? So there are instruments, there are all kinds of measurements and things that need to be taken care of. And a lot of those also are at risk of radiation. And radiation, space radiation is considered one of the most difficult and the highest risk for space exploration. And in particular for human space exploration, there are many risks that we can mitigate and try to control, but having had this sort of layered approach would be easier. So some of the most important risks that we have for radiation are the risk for carcinogenesis, and all of those could be solid organs or leukemias. The leukemia is very fluid, obviously, but some other organs are more sensitive to others. Then the risk of acute radiation, and then cardiovascular diseases, and then other processes. The problem that we have with radiation is we cannot irradiate normal people, right? It will be against the law. So what we need to do is do some sort of extrapolation or assessment with different patients that had chemotherapy or accidents that have happened, and also a lot of testing in animals. So what has happened though is we got this big grant from NASA to explore mostly based on radiation and radiation prevention methods. And then what happens though is this project was funded for reveals, and then through many collaborators, we here at UCF, we've been working on parts of it. So the funding comes from NASA, most of it, and then we have NASA CERVIC, which is the Solar System Exploration Research Virtual Institutes, and then NASA for some of the projects, the smaller projects from Florida Space Grant Consortium. All of that is focused on radiation and radiation mitigation. And as you see on the upper quadrant, the different risks for humans, then the risk for radiation is different for everybody, and also different if you're a woman or a man, because the different tissues that we have in our system, mostly obviously for women, is the reproductive system. Some of these projects, some of this information that I'm going to share with you has been submitted already to NASA as conclusions. Some of that cannot be disclosed because of export control, but then a lot of that is going up the chain of command right now as we speak, with NASA going into the moon with the lunar mission Artemis. And as we saw this morning, there was this Crew 2 that launched on the SpaceX. That was really exciting. So that's part of the overall infrastructure that is going to be assembled for operations later on going to the moon. And so we had to test all of this with NASA, and then also with the high radiation beam that we have access to, which is at NSRL, and it's NASA Radiation Laboratory that it's in Brookhaven National Lab. But I'm going to focus on two aspects of this big overall research. One is the human performance, and we are trying to see if part of the polymers that we are developing in part of the projects that we are working, how we can assemble all of that and create some prototype for radiation protection. And as we know, the the best molecule to protect for radiation, especially the high energy radiation, which is the solar galactic cosmic rays, so GCRs, is water. So right now we have a lot of polymers that have a whole lot of hydrogen components in it, but then also how we can do the simulation. If you see my pointer here, how this vest that we have here is called the cooling vest, with this tubing that is attached to it, can attenuate some of the radiation. And this is, we've been doing this to test for the thermal effect right now first, because anything that you put in the body creates heating. And if you're working and all of this exercise that astronauts will have to go when they go outside and they pick up rocks and they do walk around, it's not going to be easy. And they have a thermal overload that has to be eliminated. But this system that they wear underneath is the cooling garment can be redesigned in a way that can help also mitigate for radiation. And so we have the polymers, the chemical sort of produced new polymers, but then also the natural water that is used in the cooling system already to help for radiation protection. And so one of the collaborations I'm seeking right now is how we can redesign this cooling vest to increase surface area. And I know Dr. Yoav Peles invited me to a talk that he prepared for NSF workshop. And I presented some of this work because he's an expert in thermal. And I like also to ask if anybody in engineering would be happy, I'd be happy to collaborate with them to redesign with the students, maybe a student project, to redesign the tubing that has to go with this space vest, and then contain and continue to have that cooling effect. And then we'll layer all of these other composites. And I also have worked with Dr. Caitlin Crawford from engineering, which I know she's in here attending this talk. And we probably will put some of our sensors here to determine the loading, the heating load that we have. And the other side of my team, I am the deputy for the whole Reveals NASA Bigger grant, is also working on synthesizing this composites, which is the ones that we are going to add into this vest. Now we have the baselines, and now we have all of these composites already made, which is starting just by molecules. It's really amazing that they did this work at Georgia Tech. And then we'll add these layers on top to create a layered effect for attenuation, for radiation attenuation. And as you can see this on the right side, this type of composites, and doing the testing, they also had to do radiation at Brookhaven. We did the radiation testing at Brookhaven and also at Notre Dame. And the other side of the team, which I also, I'm the deputy, but we have working at NASA Johnson Space Center, they are working on the application of this also active dosimeters that will measure the radiation real time, not just after the fact, so the real time. So when astronauts are in operations, they can understand if there's going to be higher risk or lower risk, and then mitigate as they go. And this has to be displayed or incorporated in the spacesuit in a way that is easy for them and it's not intrusive. And the way has been already been tested at NASA Johnson Space Center for the underwater training, which is called the Neutral Buoyancy Lab, or abbreviation is NBL. They like to have these displays that are flexible, which are shown here on the left side, that are flexible, that can be attached to the helmet, where the astronaut can just scan quickly and see how far this radiation dose, or how far is it going, and then stop or call operations, and then do this sort of activities in preparation for any type of contingencies. Before, what they do right now, they have some testings, they have some dosimeters that are active dosimeters and doing some measurements, But until now, what they do is passive dosimetry. So you just get the dose, and then after that, then they tell you you can't go back anymore, or maybe you're done with your career. So there are career limits and other things. But at the molecular level, we want to study the cells, right? So the cells that we're studying and the tissues that we are studying are mostly focused on the high sensitive and the ones that are more relevant for operations, also for humans. And in this case, the main organ, obviously, that is difficult to create radiation attenuation is the brain. So neural is difficult, and it's also something that you want to preserve, right, as much as you can. Then the next, besides that, obviously, is the heart. But we have a lot of research. They have done a lot of research in cardiovascular diseases and cardiovascular damage through radiation, and there's a lot of data. But what we want to do right now is also do this, and I'm doing this in collaboration with Burnett College of Biomedical Sciences with Dr. Masternak, and also Dr. Kunal Mitra from FIT. So both of them, one is bioengineering and the other one is a biomolecular expert. I am in the between, they can see the overall aspect of it. And so what we do is we take these cells, right? Some of the cells from different tissues, and mostly we started with animals, but then later on we can get human cells, which are iSPC cells. And these somatic cells, you can see them in these constructs that are shown on the right side. Obviously, it's difficult to see them on the naked eye, but you can see the reddish tone of the gel and the cells are going to be added to it. And then you can run a fluorescence imaging to see how the cells are distributed through the gel, and then later on do the testing. So some of the testing that we do will be for oxidative stress, also for some other type of enzymatic and from inflammation processes. So the problem with the space exploration is it doesn't happen in 1G, right, at Earth's gravity. So we have to incorporate the lack of gravity or the gravity that's going to be on this planetary surface. And in this case, if you go to the moon, it will be 1/3 of the G, but 1/6 of the G of gravity of normal Earth. And then if on Mars is 1/3. Obviously, it depends on the mass of the planetary surface. And to simulate part of that, Kennedy Space Center has these facilities that we have gone to, which are these microgravity simulation devices. And these devices spin in different axes, simulating free flow, they're in suspension. The problem with that is that as soon as you stop, they go back to the sediment, right? So the tests that you have to run have to be very quickly. And then access to these facilities is not easy because a lot of researchers like to do this type of work, and the schedule is difficult to get in. But we have some good results that we have presented. Also, we are actively publishing, and continuing to move forward with all of this work. And we are going to, we got a seed funding for this microgravity biomanufacturing space activities for cells. Because in 3D vasculature, 3D constructs, it resembles more the architecture of what the real organs are, just if you were sitting on a cultural surface, right? In a traditional cell culture, it's 2D, but then 3D gives a brain structure or the structure of the organs that you want to study. So I'll end up with this, and I have time for discussion and some of the whatever questions you will have. The acknowledgements will be for the NASA funding that we have, and the collaboration with other NASA centers, and the Cell Link industry partner.